So we all know that humans perceive the outside world through their five senses: hearing, vision, taste, smell, and touch. Senses are controlled by perceptions that are received through receptors such as chemoreceptors, nociceptors, thermoreceptors, mechanoreceptors, photoreceptors, and much more. These receptors receive impulses, allowing us to perceive sensations in the world around us. These senses can be categorized into three separate groups: the exteroreceptive senses, which are associated with changes on the body surface; visceroreceptive senses, which are associated with the major organs; and proprioceptive senses, which are associated with muscles, tendons, and in-body position. Although we understand how these work, what if we were able to expand on the capabilities of each sense? Research has shown that people, under very rare circumstances, have developed extraordinary applications of their five senses, either through acquired skill or genetic mutation. Let's look at the first sense we will be covering: hearing. The way one perceives noises can be explained by the innate mechanisms of the human ear. First, the external ear captures sound waves that enter the ear canal, also known as the external acoustic meatus, which causes the eardrum to vibrate and the bones within the inner ear to move. This bone system is comprised of the malleus, incus, and stapes, and taps on the cochlea, in which sound waves trigger nerve impulses. The impulse then travels down the vestibulocochlear nerve, allowing the brain to perceive the noise. People have been able to train the ear to use echolocation, which involves emitting a sound from a source and listening to its deflection upon objects to determine the user's location. A primary example can be seen in nature in bats and dolphins. Though humans won't be able to utilize echolocation as well as animals, the ability to do so is extraordinary nonetheless.
At first glance, this may seem like an ordinary man out for an ordinary bike ride. But look and listen a little more closely. Daniel Kish is completely blind. In fact, he doesn't even have eyes, save for prosthetic ones. Daniel has never seen a tree, a car, or even the bicycle he's riding. So how does he know where he's going? Or how to avoid oncoming traffic? The answer can be found in the clicking sound you hear. It's called echolocation. Bats use it to fly around in the dark, and dolphins use it to navigate the oceans.
The second sense we'll be covering is vision. How exactly can we see and perceive the natural environment around us? Simply put, all vision is based on the perception of electromagnetic rays, but in order to better understand this process, let's explore a little more. The cornea refracts light rays into the pupil, causing the pupil to enlarge or dilate in order to regulate the amount of light passing through. Once this happens, the lens changes shape of the light ray so it can focus onto the retina, which is in the back of the eye, along with the millions of light-sensing nerve cells, called rods and cones. Cones are concentrated in the center of the retina in an area called the macula. When there is bright light, cones can provide clear, sharp central vision as well as detect colors and fine details. The rods, on the other hand, are located outside the macula and extend all the way to the outer edge of the retina. They provide peripheral vision and help the eyes detect motion in dim lighting. All of these rods and cones help convert light into electrical impulses, in which the optic nerve sends impulses to the brain and produces an image based on those impulses. Due to the substitution at codon 180, a genetic trait has developed called tetrachromatic vision. This enables an individual to see 4 primary colors instead of 3, since the mutation causes another type of cone in the retina to develop.
And only see 10,000 colors, is it also possible to have 4 different cones and be able to see 100 million? That's what Newcastle University neuroscientist Gabrielle Jordan has been searching for decades, and in 2010, she finally found one. Jordan was testing women who have colorblind sons, the rationale being the sons must have inherited a mutated X chromosome from their mothers, so the women must have 2 different X chromosomes and therefore 4 cones in their eyes. The women looked at 3 colored circles that to most people would look identical, but one had just a little red or green mixed in and would only be visible to someone with 4 active cones. And out of the 24 women who took the test, only 1 got every single question right. So it's rare. It's estimated only about 1% of people have a 4th cone, and even then, many don't seem to use it. And even when they do, how can they communicate what they see to us 3-coned non-superhuman?
The third sense that will be covered is taste. Normal people are able to detect 5 dominant flavors: sweet, salty, bitter, umami, and sour. Taste is perceived by a number of receptors, including chemoreceptors, thermoreceptors, mechanoreceptors, and nociceptors, each contributing to the perception of food. Chemoreceptors are able to detect certain chemicals in food and drink. The water must be soluble in order for taste to work properly. In fact, these receptors require direct contact with food and do not adapt quickly to taste, whereas the other receptors have a less dominant role. Thermoreceptors detect spicy foods and methanol, nociceptors also detect spicy foods, and mechanoreceptors detect the texture of different foods. Although 80% of taste is controlled by the smell of different objects. Advanced taste can be seen today through supertasters. These individuals have two dominant alleles for the gene TAS2R28, which allows people to perceive a stronger sense of taste, specifically with foods too bitter, sweet, or spicy. The primary difference between supertasters and non-tasters is that supertasters have more papillae. Continuing on, smell has the ability to make people think of memories, just by the familiarity of an odor. How we perceive an odor, on the other hand, is a process in which it begins with odor molecules dissolving into mucus at the top of the nostrils. Underneath the nostrils, in the olfactory epithelium, receptor cells called the olfactory receptor neurons detect the odors, in which they will then transmit the information to the olfactory bulbs that are located in the back of the nose. Finally, the receptors on the olfactory bulbs will send the information to the brain, allowing us to perceive and interpret this information. Throughout various environments, people have observed various species that have the ability of advanced smell, specifically the canines. This is due to the amount of neurons that are associated with a nerve. Typically, humans have 10 to 20 million neurons associated with olfactory nerves. So if we are able to increase that amount through DNA analysis and reconstruction to 100 to 200 million, causing a 10 times more sensitive sense of smell. Touch is controlled by a variety of receptors, including free nerve endings, Meissner's corpuscles, and Pacinian corpuscles. Free nerve endings are simple receptors in an epithelial tissue that detects an itching sensation on the skin. Meissner's corpuscles, or tactile corpuscles, are small flattened oval masses in connective tissue that detect fine touch on areas of the skin, such as lips, fingertips, palms, soles, and many more sensitive areas. Pacinian corpuscles, or lamellar corpuscles, are large episodial structures made from connective tissue fibers located in the deep dermis of hands, the urethra, breasts, tendons, and ligaments. Lamellated corpuscles are able to detect heavy pressure and vibrations. Other than mechanical feelings, there's also the feeling of temperature and pain. Temperature extremes are detected by thermoreceptors, while pain is detected by nociceptors. There is much talk of having a sensitive touch, but having true sensitive touch to the point that is unreal can happen through genetic changes. The gene ATL1 provides instruction for providing a protein, elastin-1, which is present in the brain and spinal cord. It activates axon growth and it directs the growth of specialized extensions. We can genetically change someone's DNA by increasing the amount of elastin-1 via ATL1. You can increase sensitivity. With the sensitivity comes the side effect of increased pain. To counteract this, we will intentionally induce congenital analgesia, a genetic mutation of the SCN9 gene affecting the build of sodium channels on nociceptors, causing reduced to no pain. This can allow one to have near microscopic touch without the consequence of pain. Many of these changes can be made through genetic modification, mainly through CRISPR and designer babies. Designer babies are embryos that have had genetic modification during the process of IVF, also known as in vitro modification. This is a process in which the egg is fertilized in a lab and grown there for about a month, which is then artificially inseminated into a female to allow the child to develop until birth. During this period, scientists can analyze the DNA and even now change the eye color of the kid or a single gene trait. This can allow someone to change a child's gene sequence before birth legally, even though the concept of designer babies is largely controversial. CRISPR and Cas9, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a device used for editing genomes that act as a molecular pair of scissors on cutting DNA. It is an RNA-guided site-specific alteration that can allow one to change the precise sections of DNA. Both methods of gene alteration can make these supersenses a universal trait.